For these lessons, we're going to learn microtubule organizing center, or known as MTOCs. Okay, so what's mean MTOC? MTOC. Okay, stand for microtubule organizing center. So it means that we're going to look at in detail about microtubule. Okay, so the microtubule organizing center is a structure. Okay, it's not an organelle. Highlight. It's not an organelle, guys. It's a structure found in the eukaryotic cell from which microtubule emerge. So means that if you want to form microtubule, okay, or you want to organize a microtubule, any organelles can perform, or any structure that can perform this role will be termed as MTOCs. Are you clear? Okay, so means the MTOC is not organelle. It's not like chloroplast, one organelle, mitochondria, one organelle. No. Is any organelles that can perform. The organizations of the microtubules will be termed as MTOC. So it depends on different organisms. For example, what we learn in animal have MTOCs. So animal MTOCs consist of these two important MTOCs called basal body, which is associated with cilia and flagella, and centrosome, associate with the spindle formation. Are you clear? So for animal, our MTOCs, basically we have two kinds of MTOCs, means that two different organelles can carry out these microtubule organizations centers role. One is a basal body involved in the cilia and flagella. Later we'll look at it, how they look like, and the centrosome associated with the spindle formation. So I'm going to explain what is spindle and spindle microtubule, okay, later. But how about the yeast cells? Yeast cells slightly different. Yeast cells, they don't have the centriole. Can you see that? They don't have the basal body. They do have different way to actually organize their spindle. So they use the nuclear envelope. In plant, they also use the nuclear envelope itself. So these two are not in our syllabus, just for information only. So to tell you that different organisms, they have the different organelles to perform the MTOCs. But what we need to know for animal, we need to know basal body and also the centrosome. Okay, these two will be involved in the organizations of the microtubule. So we start with the centrosome. Okay, we start with centrosome. Centrosome, write down, they are non-membrane bound organelles. They don't have membrane. Okay, non-membrane bound, and they only appear in animal cells. Okay, only animal cells. So this centrosome, if you look at this, they don't have the membranes. They don't have membranes. So what is this? So they have the protein that's surrounding what? They have the protein that's surrounding the centriole. So this one is the centriole. So from this structure, you can see that microtubule actually emerge. Can you see that? Microtubule actually emerge. So the center part here is a centriole surrounded or packed together with the protein. So this protein is known as the pericentriolar material or pericentriolar matrix. Can you see that? So the entire structure is known as the centrosome. Okay, so if we label, so this structure is known as the centriole. Can you see that? Centriole. I will explain more about the structure of centriole later. Okay. Next, surrounded by what? Surrounded by the pericentriolar material. Okay, pericentriolar material. Okay, so the entire structure here actually involved in the formations of the microtubules. Can you see that microtubule come out from this area? Come out. Right. So this one is termed as spindle microtubule. Okay, spindle microtubule. Okay. So the centrosome have three important activities. So what they do, they actually nucleate the polymerization of tubulin subunit into the long polymer that are microtubules. So another words here means that centrosome form microtubule. Okay, centrosome form microtubules. Who helps them to form this microtubule? The pericentrolar material. PCM, pericentrolar material, is the one that helps to polymerize it. Okay, but after I form this microtubule already, I want to make sure it is functional or useful array, then centriole will help to organize. So this is done by centriole. So centriole actually organize this microtubule into the useful array. Okay, so students always confuse. Okay, students always confuse the role of the centrosome versus the role of the centriole. So let me show you something here. Okay, a quick one. Now, we do have this. Okay, let me show you a quick analogy. Okay, a quick analogy. So you have a factory. Okay, you have factory. So this factory actually, okay, making t-shirt. Okay, making t-shirt. So it's a t-shirt factory. But in order to make a t-shirt, you need a raw material. So this raw material, they are the thread. So it means that one part of the factory form the thread. Okay, so form the thread. But my factory is not selling the thread. My factory actually selling t-shirts. So another compartment, another area of the factory, okay, take these t-shirts, take the thread, form the t-shirt. Can you see that? Are you clear? So same goes to our centrosome. Same goes to the centrosome. So centrosome, the entire centrosome consists of two things. One is the centriole. One is the centriole, okay, the blue color centriole. Another one actually is the pericentrolar matrix. So what's pericentrolar matrix do? Pericentrolar matrix, the protein here actually help us to form spindle microtubule. Are you clear? It forms spindle microtubule. But this spindle microtubule is something like a thread. No function. It's just a thread. It's just a spindle microtubule. So we need to organize it. So who organize it? So centriole will be the one that organize. Organize what? Organize the spindle microtubule. So they become a useful array. So this useful array, so they become useful array in a useful manner. So this useful array actually is termed as the spindle. So what's the difference between spindle microtubule and spindle? Right? Spindle microtubule and spindle, actually, they are not the same. So during the mitosis, we learned that centriole actually located at the two opposite poles. Okay, for those SPM students, you should be able to know about this. Okay, so this spindle, okay, so where is the centrosome? Centrosome will be somewhere here, the centrosome. So centrosome actually produce a microtubule. So this microtubule will be arranged by of the centriole so that it will form this spindle. Can you see that? Continue. So it form. Can you see that? So if I label this one by one, we call it a spindle microtubule. Okay, then entire structure, okay, here. Entire structure is known as spindle. So means that we arrange a spindle microtubule to form spindle. Or spindle microtubule is the same as spindle fiber. Spindle fiber is what you guys use in your SPM. Okay, but in A level, we use the word spindle microtubule. So be careful, spindle microtubule refer to this microtubule. Okay, refer to this microtubule. So we have how many? We have 5 spindle microtubule, something like a thread. So we have thread. Okay, so this thread, we have to organize them, right? So we make it into a t-shirt. So the entire thing is known as the t-shirt. So spindle is a t-shirt, spindle microtubules is a thread. Are you clear? Okay, so understand now. So be careful when you write. So that's why always confused. What is the role of the centriole? The role of centriole is to organize spindle microtubule so that it form spindle. But you cannot write centriole form spindle microtubule because form spindle microtubules is done by pericentriolar matrix. Are you clear? So let me put it in, the summary here. If a question asks you what are the roles of centrosome? So centrosome is entire factory, right? Entire factory. So what they do? So first, so means that the components, okay, will contribute. Like for example, in the college, so we have pre-U programs, right? So we have how many pre-U programs? We have 5 pre-U programs. Okay, so A-level lecturer only teach A-level. MAFI lecturer only teach MAFI. Can you see that? So what is the role of A-level lecturer? A-level lecturer teach A-level. That is a role specific. But what is the role of pre-U lecturer? Or pre-U lecturer teach A-level, teach MAFI. So it's a collective, So role of centrosome, first, understand, huh? So role of centrosome, first, formations of spindle microtubule. So in charge by who? In charge by pericentriolar material. Second row, organized. Okay, spindle microtubule. Okay, into spindle. So this row is done by centriole. So collectively, these two will be the role of the centrosome. Okay, but if the question asks you what is the role, general role of centriole, because when we learn about mitosis, then we have to add on some else. Okay, so role of centriole is very simple, only one row, organized. Spindle microtubule to form spindle. So there is always a lot of times questions, particularly in paper 1. So they say, in other words, also spindle formation. Correct. In other words, we can say as a formations of the spindle, not spindle microtubule. Spindle microtubule is not the role of the centriole, but it's the role of the centrosome. Are you clear? Okay, so be careful. Or the role of the pericentriolar material. So are you guys clear now? Students always confused about these two. Centrosome, formation of the spindle microtubule. And then we organize the spindle microtubule into spindle. Okay, that is the role of the centrosome overall. But if you zoom in into detail about centrosome, as a centriole only have one role, to organize the spindle microtubule so that it can form the spindle. Okay, so this, in other words, also known as the formation of spindle or spindle formations. So involved by, done by the centriole. Okay. So now next, how is the structure of centriole? Okay? Most of the centriole are made up of nice set of microtubule triplets, highlights, arranged in what we know as 9+0 arrangement. Okay, 9+0 arrangement. Okay, so this one only form one centriole. 9+0 of them only form one centriole. So in fact, per cells, we're going to have two or a pair of centriole where they're going to align at the right angle to each other or perpendicular to each other. Are you clear? Okay. So the centriole usually reside in a pair with the cylindrical centriole at right angle. the duplicate of centriole happen in the interface of the nuclear divisions. This is not really correct. Okay, so interface of, guys, cancel my mistake. Interface of cell cycle, more appropriate. Okay, change the note a bit here. So in the interface of cell cycle. So the main function of centriole is to organize, okay, organize the spindle microtubule so that to form spindle. Here, so make it clear. Okay. So now what is meant by these microtubule triplets? Okay, so microtubules, we know that one microtubule is like this. Okay, they are hollow tube like this. So we do what we do. We actually take 3 of them, group them together. So this one is called as the microtubule triplet. Are you clear? These microtubule triplets. So we total have how many of them? We use 9 of these to form one centriole. So if you look at the diagram here, you realize that here, in this case, 1, 2, 3. Can you see that? 1, 2, 3. This is one triplet. Okay, use another color. Maybe red. Okay, this is one triplet. So 2 triplet, 3, 4, 5, 6, 7, 8, 9. 9 of them. Can you see that? We arranged 9 of them. Can you see that? So 9 triplets to form one centriole. And center is hollow. Nothing at the center. So this arrangement, why we call 9 plus 0? 1, 2, 3, 4, 5, 6, 7, 8, 9, center, nothing. So we call it as a 9 plus 0 arrangement. Okay, 9 plus 0. So 9 refer to what? 9 refer to the triplet. And then this one form one centriole. So we another centriole. Can you see that they are perpendicular? So how we actually identify them later? Okay, in the photomicrograph or sorry, electron micrograph, then you can see that because they are perpendicular to each other. If you look at this diagram again, so can you see that 1, 2, 3? So we have 1, 2, 3, 4, 5, 6, 7, 8, 9. So this one is the, if you look at this is what we call the transverse sectioning, round shape. Why rod shape? This is the longitudinal sectioning. So if you look at the diagram here, so if I have the rod shape like this, so they are perpendicular to each other. There'll be something like this. Okay, perpendicular to each other. So if I view it from here, you can see that this one will give us the round shape, then this one will give us the rod shape. Can you see that? You view it from that angle. So therefore, how to identify? First thing, you want to identify whether they are centriole or not. You look at this. They must be perpendicular to each other. So it means that one round, one rod. And then we have the 9 triplets and center hollow. So this one must be the centriole. Are you clear? This one must be a centriole. So arrange in a 9 plus 0 arrangement. Okay. So now label. So you can see that this is the centriole pair. Pair of centriole consists of the microtubule triplet. So total we have 9 of them to form this microtubule triplet. Okay, so another ones or another organelles, okay, or structures that formed by microtubules, they are cilia and flagella. Okay, so cilia and flagella. So eukaryotic cilia and flagella, they are motile. So basically, they are movement. They can move. Are you clear? They can move. In terms structurally, they are quite identical. Okay, but in terms of arrangement, slightly different compared to the centriole. They are in the form of 9 plus 2 arrangement. So how this 9 plus 2 arrangement is special? So just now in centriole, okay, in centriole we learned about 9 plus 0. The 9 is the microtubule triplets, but here is the doublets. So means that we have one microtubule. We join with another microtubule. So this is called doublet. Are you clear? So these doublets, we have 9 of them forming the outer ring. So outer ring, we have 9, okay, set of microtubule, okay, doublets. And center, we have 2. They didn't join together. So centers here, we have 2 single microtubule. So therefore, we call this as a 9 plus 2 arrangement. Are you clear? 9 plus 2. The 9 here refer to the microtubules doublets surrounding the 2 central single microtubule in 9 plus 2 arrangement. And the interaction of this, can you see that? The interaction between these dying proteins as well as the central and the outer microtubule allows the movements or beating action of the cilia and flagella. Okay, so who organized them? Who organized them? Like, I mean, forming this a basal body. Okay, basal body act as the MTOCs. Okay, basal body is another MTOCs that organize the spindle microtubule to form the cilia and flagella. So we have to look at the structure of cilia and flagella. So structurally, they are identical. They are arranged in the 9 plus 2 arrangement with 9 sets of the microtubule doublets surrounding 2 central single microtubules. Okay, so now, stop recording. Okay, so for this structure of cilia and flagella, what you need to know, so because they are arranged, okay, in 9 plus 2 arrangement. So what's been 9 plus 2? So you can see 9 doublets, right? 2 single microtubules. So here is the outer microtubule doublets, or just say microtubule doublets. Okay, either say outer microtubule doublets or just microtubule doublets. Okay, so this one's the inner one. So the central single microtubule or just central microtubules, okay, or single microtubules. And we know that the structure here that's organized this microtubules, not spindle microtubules. Okay, be careful. Basal body, nothing to do with the spindle. Okay, so basal body here is the one that organized the microtubules to form the cilia and flagella. So here answer is the basal body. Okay, it's the one of the MTOCs in animal. So my question here is, plant cell has cilia and flagella or not? Yes, what is the answer? You can type it out. Plant cell, do they have cilia and flagella? The answer is no. Okay, they don't have cilia, but one kind of plant cell they have the flagella, but it's different kind of flagella. Are you clear? Okay, so yes, only in mammal, also mammal, in the animal, sorry, in the animal, only animal have cilia. This animal cell have cilia and flagella, not all, some of them. Okay, so let us look at where we can get the cilia and flagella. Okay, so first of all, let us look at this. So if you look at the structure, you realize that here 1, 2, 3, 4, 5, 6, 7, 8, 9. Can you see that? 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 doublets and 2 single microtubule. Can you see that? 2 single. So this one either cilia or flagella. Either cilia or flagella. Then we look at the density. If many of them, then this one must be cilia. So that's why this is the transverse sectioning. That's why I see the round shape because of the longitudinal sectioning. Can you see that? So many. So that's why many, they are cilia. Okay. But if you look at this, Chlamydomonas with the flagella. Can you see that? With the flagella. So again, you can see that 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 plus 2, 9 doublet with 2 single microtubules. So this one, okay, in terms of density, not so many, 2 of them only. So this one most likely will be flagella. But if I show you only 2, it's very difficult for us to actually identify if it's 2 only. So it might be cilia, it might be flagella, because in terms of structure, they are very, very similar. Okay. So let us look at the comparison between the cilia and flagella. Now, in terms of definition, cilia, so cilia are short, hair-like appendages extending on the surface of a living cell. So it must be animal cells. Flagella are long, thread-like, okay, long, thread-like appendages on the surface of the living cell. So basically, cilia shorter, flagella longer. In terms of density, so you can see that cilia high in terms of density. So many, even hundreds of them per cells, okay, for cilia. Flagella, less, okay, in terms of density will be lower, about 10 per cells max, okay, less than that, okay. In terms of functions, cilia actually more involved in the lining of the trachea, where they sweep the mucus and dirt out of the lung. Or in female mammal, the beating of cilia in the fallopian tube or oviduct move the ovum from the ovary to the uterus. Okay, this is where you can find the cilia. So basically, they involve in the movements of the substances. Flagella is the movement of the cells. So for example, in terms of cell motility, so the sperm cell which uses flagella to propel itself through the female reproductive tract, okay, the tail, we always say the tail, right? So this one actually is the flagella. Okay, so you need to know this, guys, to differentiate between cilia and flagella. So I repeat again, for cilia and flagella, what we need to know. Summary. So first, cilia and flagella in terms of structure, they are identical. Okay, all of them arranged in a 9+2 arrangement. So they have the microtubule doublets surrounding two single. So how many microtubule doublets? 9 of them. Okay. So who actually make the cilia or organize the microtubule to form cilia or flagella? So the MTOCs. So the name of MTOCs here known as basal body. Okay, basal body. So in terms of structure, you should know that, okay, the cilia many, flagella less. Okay, not so high the density. Okay, but how you compare between both of them? So we compare among the definitions, structure, density, as well as their functions, different function. Okay, but if you compare them with the centriole, definitely easier, because centriole always 9 plus 0. Okay, centriole always 9 plus 0 arrangement. Okay. So do you guys have any questions? I don't have the record now.